Alright, so this is the third lecture of the molecular biology unit, and in this lecture we're going to be talking about transcription. So gene expression begins with the process of transcription, which is copying the DNA into an mRNA strand. And then the mRNA strand leaves the nucleus and comes down here and is translated by the ribosome and the cytosol into the protein sequence. And so if we show here, transcription is similar to DNA replication, except in this case, you're copying the DNA strand and synthesizing a new RNA strand. And you can see the uracils here. So RNA is single-stranded, and it's a molecule that can take on 3 different forms in the process of protein synthesis. And the 3 forms are messenger RNA or mRNA, transfer RNA or tRNA, ribosomal RNA or rRNA. We'll talk about each of these individually over the next few slides. mRNA is shown here, messenger RNA, and that makes sense because it carries the message of the gene that's being expressed from DNA, and then it leaves the nucleus and it can be translated into the protein sequence. So the messenger RNA, like we said on the last slide, it serves as the template for the amino acid sequence. An important distinction to make between prokaryotes and eukaryotes is that in prokaryotes, this mRNA that's generated from transcription is the final product, and it can immediately undergo translations. So it can leave the nucleus and undergo translation. However, in eukaryotes, you produce this mRNA transcript, and then it has to undergo further processing. And what that really involves, and we'll talk in much more detail about this in a few slides, is that so in an mRNA sequence in eukaryotes, you have what's called exons and introns. And the introns are the non-coding region, so you have to remove those. And then you got to connect the exon sequences, and that's your final mRNA sequence in eukaryotes. So you generate that, and then that can leave the nucleus and undergo translation. So transfer RNA or tRNA, we'll talk in much more detail about these in the protein synthesis lecture, but just to give you a background, here's a tRNA here, and you can see it where it carries amino acids that are in accordance with specific 3-base RNA sequences called codons. And again, we'll talk in much more detail about that when we talk about protein synthesis. So they come in here to the ribosome, and they're carrying this amino acid that corresponds to a codon in the RNA sequence. And then eventually the ribosome keeps transferring these amino acids to form a nice polypeptide chain. And then lastly, ribosomal RNA or rRNA. This combines with proteins to form ribosomes. So it's part of the ribosome structure, and then it helps carry out translation of mRNA. And the other thing with rRNA is that it's synthesized in the nucleolus within the nucleus. So the major player in transcription is an enzyme called RNA polymerase. And this exhibits multiple functions to carry out the process of transcription. First off, it unwinds the DNA double helix at the transcription start site, which is analogous to helicase in DNA replication. So you can see that here where it's unwound the double-stranded DNA, and then it goes on to read the DNA sequence and generate the RNA strand. So you can see that here where it's moving along, generating the new strand. And the same convention holds true. It reads the template strand in the 3' to 5' direction, and then it generates the RNA strand in the 5' to 3' direction. The other thing is, as RNA polymerase is moving along the strand, the DNA alpha helix reforms. Now it elongates in the 5' to 3' direction by adding ribonucleoside triphosphates to the free 3' OHN, which is very similar to how DNA polymerase elongates a DNA strand. As a result, just like in DNA replication, the RNA strand, it produces a new 3' OH for the next base to be added on. Now, a couple conventions we want to go over. Now, we apologize. There's a small error in your book. Please correct this. Now, the strand of DNA being read by RNA polymerase is called the template or antisense strand. We have that labeled here for you. So here's the antisense strand of the template strand, serving as the template for the newly synthesized RNA, which is in green here. And remember, the antisense strand of the template strand is complementary to the mRNA being generated. Now the sense, or the coding strand, which is up here, this strand is complementary to the antisense strand of DNA, which makes sense, but it's the same sequence as the RNA being generated, except for the fact that it will have thymine instead of uracil because it's DNA. So once again, one more time, RNA polymerase reads in the 3' to 5' direction. On the template strand, also known as the antisense strand, it generates RNA in the 5' to 3' direction, which is complementary to the antisense strand and is the same sequence as the sense strand. Now, eukaryotes, they contain 3 different types of RNA polymerase, and this is important to remember. So RNA polymerase 1 produces rRNA, or ribosomal RNA, and this is the most abundant type of RNA. RNA polymerase 2 produces mRNA, which is the largest type of RNA by size. As we showed on the previous slide, RNA polymerase 2 has the ability to unwind DNA at the transcription site, also known as the promoter, which we'll talk about the promoter in a few slides. And then RNA polymerase 3 produces transfer, or tRNA, which is what's involved in translation, like we talked about, and it's the smallest type of RNA by size. Now, none of these types of RNA polymerase possess the proofreading ability, unlike DNA polymerase, which does possess proofreading ability in the process of DNA replication. Prokaryotes are much simpler. They only have one type of RNA polymerase, and it produces all types of RNA. So it produces rRNA, mRNA, tRNA, all of them. Now, what's interesting is that the antibiotic rifampin inhibits RNA polymerase in bacteria specifically, but it does not affect RNA polymerase in eukaryotic cells. All right, so reverse transcriptase. This is an enzyme that catalyzes the synthesis of DNA from RNA. And this is an enzyme that's not found in humans. It's typically found in viruses, most notably the human immunodeficiency virus, or HIV, among other retroviruses. And they utilize this reverse transcriptase to synthesize a DNA copy of their RNA genome. So we'll show you in the diagram here, this is the host cell that's infected by the virus. The virus comes in like this, so this is our virus. It has double-stranded RNA, which is also unique. So this double-stranded RNA is copied by reverse transcriptase and converted into double-stranded DNA. Now the reason for doing that is so that then this double-stranded DNA, this viral DNA, can then get integrated into the host cell DNA, and then the host cell DNA replication machinery can actually then replicate the viral DNA, which ends up turning the host cell into a viral factory. And so it creates viral proteins, replicates the viral DNA, and then you form new viruses that can then go on and spread the infection. And that's part of the reason why these viruses are so devastating. All right, so now we're going to talk a little bit about gene structure and how it relates to the transcription process. So first, prokaryotic gene structure. Prokaryotic genes are preceded by promoters. So here you can see that in the sequence here. These serve as the initial site where RNA polymerase binds and begins transcription. So you see RNA polymerase that's binding the promoter, and then it goes on to read the template strand of DNA and synthesize the strand of RNA. Now what I would note here, just to make this a little more clear, is RNA strand synthesis, because we're talking about here, proceeds in the 5' to 3' direction. Remember, it's red in the 3' to 5' direction. So RNA polymerase moves down this template strand in the 3' to 5' direction, and then when it reaches a sequence called the chain termination sequence, which is shown here in red, the newly synthesized strand is actually forming what's called a hairpin loop. And you can see that here in the diagram. So you see some base pairing here that results in this loop-like structure. And what this does is it creates this bulky structure which then dislodges RNA polymerase from the DNA. So it terminates transcription of this gene. Eukaryotic genes, they're also preceded by a promoter, and this is where RNA polymerase 2 binds. So if we put a nice little 2 in there. Now what's important to know is mutations in the promoter sequence, they can result in significantly decreased gene transcription, and that makes sense because you can't even begin the process of transcription if you don't get RNA polymerase bound to the DNA sequence. So then as a result, you have decreased expression of the gene, which makes sense. And again, it's just because promoter binding is such a key step. Eukaryotic promoters have AT-rich sequences, meaning adenine, thymine, and these serve to help RNA polymerase and other transcription factors comprise the initiation complex binding to promoter. So what you have here is you have RNA polymerase that binds, and then you have these proteins called transcription factors that also bind to the promoter and then other areas on the DNA. And the transcription factors help facilitate RNA polymerase binding and initiation. So their role is often supportive. They work in tandem with RNA polymerase to help carry out the transcription process. And then 2 sequences to be aware of is the TATA box, or thymine, adenine sequence, which you'll find in the promoter here in orange, and then the CAAT box, which is another sequence commonly found in eukaryotic promoters. And these are regions that are recognized by RNA polymerase and transcription factors to help aid in recognition of the promoter sequence and then binding to it as well. All right, so you remember we talked about a key distinction between eukaryotic mRNA and prokaryotic mRNA. Prokaryotic mRNA that's produced from transcription is ready to go. It leaves the nucleus and undergoes translation. However, eukaryotic transcription produces a primary mRNA that's also known as heterogeneous nuclear RNA or hnRNA that must undergo further processing in the nucleus. And so the reason for that is that there's, in this, unlike the prokaryotic RNA, which is fully ready to go to undergo translation, there are coding sequences called exons, and then there's non-coding sequences called introns, which are shown in gray. So you need to splice out these introns and then connect the exons together into a sequence that's ready for translation. And you'll see why that's important in a few slides. So just to establish where this is occurring, transcription produces that hnRNA. That undergoes further processing to give you the final mRNA, which then leaves and undergoes translation. So this is all happening in the cytosol, is the key takeaway from the slide. So it actually begins during translation. So during translation, the 5' cap is added and the poly A tail is added. And you can see that here. So this is your DNA. This is your RNA transcript being formed. Here's the 5' cap in the figure, and here's the poly A tail. So the 5' cap is a 7-methyl guanosine sequence that's added to the 5' end of the mRNA. It functions to protect mRNA from degradation by exonucleases. It also helps regulate nuclear export, promote 5' intron excision, and promote translation. So it's a very protective mechanism that also helps facilitate other processes. Poly A tail occurs during transcription with the addition of a sequence of only adenine bases, hence the poly A tail, to the end of the mRNA strand as the gene terminates. So when RNA polymerase is going down the gene, when it reaches this termination sequence, that's when you start adding on these adenine bases to form the poly A tail. And again, this functions to protect mRNA from degradation and then promote nuclear export. So these are two modifications that are put on either end of the RNA transcript to promote other processes and also protect it from degradation. So after you've added the 5' cap and the poly A tail, you have this hnRNA. And before the transcript can leave the nucleus, it has to undergo splicing, which again consists of removing these introns out and connecting exons together, so that the final mRNA product is a continuous sequence of exons, which is what you want for translation. So we're going to take a few slides here to go over the general mechanism of splicing. This is actually a fairly high yield topic for biochemistry exams, could be seen on a board exam as well, so you want to be aware of the major players here and what's happening. So you have small nuclear ribonucleoproteins, also known as snRNP, that assemble with other proteins to form the spliceosomes at splice sites. And you see that with these green ovals like this, and they're forming at the splice site. Now a splice site is probably what you're imagining it is. It's the junction between the intron and the exon, so where the cut is going to happen. Now what the figure shows nicely here is you have a 5' splice site, which is on the 5' end of the intron, close to the 5' end of the transcript. And you have a 3' splice site, which is on the 3' end of the intron, closest to the 3' end of the RNA transcript. So after the spliceosomes have formed at each splice site, they begin pulling the exons closer together, which then causes the intron to form like this, and they're getting ready to cut the intron out. They excise the intron, and it forms a lariat, or looped structure, and you can see that here. And then they completely cut out the intron, and then they join together the two exons that were on either side of the intron to then form a mature mRNA that can then leave the nucleus and undergo translation. So splicing continues to occur across the hnRNA, or also known as the primary mRNA or the pre-mRNA. Pre meaning you're not at the mature point yet, until the transcript is composed of a continuous sequence of exons, so as we show here. We've mentioned this before, but just to remind you, the untranslated regions, which are UTR, are at both the 5' and the 3' end of the mature mRNA. So they actually make it all the way down to the mature mRNA. They survive splicing and they make it down to this final transcript, but they do not undergo translation. So these are not coding regions. Different than introns. These are very different. They're not cut out during splicing. They actually make it to the final transcript. So the process of splicing is actually a little more complicated. You have also what's called alternative splicing. So what this involves is that you can take different exons from the same mRNA transcript and form unique proteins. So what that means, so you have exon 1, 2, 3, and 4 here. So you can put them all together like this, and you can have one protein. So we'll call that protein 1. You could also leave exon 2 out, and form this one, and that could end up being protein 2. And then on this one, you could leave out exon 3 and just do 1, 2, and 4, and these guys form protein 3. So protein 1, 2, and 3 here all come from the same mRNA transcript. Now, as you can see, the advantage of this is that you can efficiently create a large number of unique proteins from one mRNA transcript. Now, where this can come into play is that you can have a protein like fibronectin, which has multiple functions. So it's the same protein, it just has some tweaks brought on by alternative splicing that result in it coming in 3 different forms. So you have an extracellular matrix form. So this is a form that's going to be interacting with other extracellular matrix cell surface proteins. So that could be this one. This could be the first protein that's formed. Then you have a separate form that's soluble in blood plasma. That's going to have a different chemical nature, slightly different function, so it's going to have a different sequence of exons. And then lastly, you have the cell surface version, which has to be embedded in the membrane, which means it has to have the correct chemical nature to interact and embed within the cell membrane and carry out that function. So the point here is that you have 3 different versions of this protein, but they all come from the same mRNA transcript, and that's a result of alternative splicing. Now, abnormal splicing variants can actually play a role in some cancers, many genetic disorders that include such as beta thalassemia, which is a red blood cell disorder. And to illustrate this on the diagram, so you have your introns here. Part of the reason the introns don't code for anything is they never make it to the final mRNA transcript. They're cut out. Let's say the spliceosome makes a mistake and cuts into the intron and brings it with part of the exon sequence. That's a problem because you just introduced this portion of the intron into the final sequence, which isn't supposed to be there. So it's going to throw off the final sequence, which then can throw off translation and throw off the protein sequence, which then could affect the protein function, and that's where disease results. Something else to be aware of is that these 2 diseases involve antibodies to SNRMPs, systemic lupus erythematosus, autoimmune disease that's actually characterized by the synthesis of antibodies to SNRMPs. So how this happens. So antibodies are created in response to SNRMPs, the antigen in this case, so they're mounting an immune response. That's where these patients get their symptoms from, is the immune response. These SNRMPs, they come from lysed cells of damaged tissue. So the tissue gets damaged, and then the cells get lysed, and they end up spilling all their contents into the blood, and then the immune system recognizes them and mounts a response. And then mixed connective tissue disease, or MCTD, is also highly associated with anti-U1 RNP antibodies. So over the next few slides, we're going to talk about the Lac Operon, which is a unit of DNA, as you can see here, that is involved in regulating gene expression of specifically these genes here, LAC-Z, LAC-Y, and LAC-A, that code for proteins that are involved in lactose uptake and metabolism. And the Lac Operon is important because it illustrates a lot of important concepts with regulation of gene expression in prokaryotes. So E. coli specifically prefers to use glucose as the metabolic substrate, but it is able to switch to utilizing lactose when it's available and glucose is deficient. That's the key thing, is when glucose is deficient. So the Lac Operon gets activated when glucose is absent, and then it switches the cell's metabolism to using lactose as the primary substrate. So first, the components of the Lac Operon. So you have LAC-Z, LAC-Y, and LAC-A. These are the genes that we're coding for, and these are the genes right here. This is the proteins they code for. LAC-Z codes for beta-galactosidase, LAC-Y codes for lactose permease, and then LAC-A codes for galactoside transacetylase. And these 3 proteins are involved in lactose uptake and metabolism. And that's the key thing. Beyond that, you don't need to know the specific function of each of these unless your professor really made a big deal about it and expects you to know it on their exam. So then you have LAC-P, which is also known as the promoter, which is shown here. And this is the region, like we've talked about for the promoters and when we talked about general gene structure, that's required for RNA polymerase to bind, and then it can go on and transcribe the genes. LAC-O, or the operator, which is this guy here, this is a portion of the unit that's involved in transcriptional regulation of the Lac Operon. You'll see where that comes into play in a minute here. Then you have this gene LAC-I, which is over here upstream. This codes for the Lac repressor protein. So what is the Lac repressor protein? In the absence of lactose, the Lac repressor is active. So this protein is active, and what happens is it comes over and it binds to the operator site and blocks RNA polymerase. So if we had RNA polymerase here, RNA polymerase can't physically go down the gene and transcribe it, and thus prevents transcription of the LAC-C, LAC-Y, and LAC-A genes. Then you have this cap site here, which is just upstream from the promoter. And the cap site is where the cap protein, or the catabolite activator protein, binds. Now in the cell, cap combines with cyclic AMP, and we show that here. So you have adenylate cyclase that's synthesizing cyclic AMP from ATP. Then cyclic AMP binds with cap, so then you have this complex, then comes down and binds to the cap site, which then facilitates binding of RNA polymerase, which is shown here with this circular ovoid structure. And when glucose is not present, you actually have an increased production of cyclic AMP, increased binding of cap to the cap site, and then stimulating increased binding of RNA polymerase to the promoter, which then facilitates transcription. However, when glucose is present in the cell and lactose is not needed, you have this Lac repressor protein. Remember this LAC-I gene? You have this Lac repressor protein, which is produced here. It's active. It binds to the operator region, which is shown here, and inhibits transcription. And in this state, when you have glucose present, this repressor protein is produced continuously. So you're continuously blocking transcription. And that makes sense. You have glucose present. You don't need to produce all this metabolic machinery to metabolize lactose because you're relying primarily on glucose. And thus, as a result of blocking transcription of these genes here, you're going to decrease production of their protein products. So another thing, when glucose is present in the cell, it's actually going to inhibit adenylate cyclase. So we'll talk about this here. So this is just repetitive. This is an error in your book. We apologize. So make sure in your book you cross this out. Glucose is present. You just have decreased production of cyclic AMP because it blocks adenylate cyclase. So you have decreased cyclic AMP production. You're going to have decreased cap binding to the cap site, and then decrease promoter binding by RNA polymerase, and then decrease transcription of the genes. Now, switching gears here, when lactose is present in the cell, you have a small amount of it. So if you have lactose here, a small amount of it's going to get converted to allolactose by beta-galactosidase. And in this case, allolactose is what's called an inducer. And this is just a substrate that's involved in promoting gene expression. So in this case, what allolactose does is it actually binds to that repressor protein produced by the LAC-I gene. And then what that does is it induces a conformational change in the repressor protein, rendering it inactive. So you don't bind. This binding to the operator does not happen when lactose is present. So then RNA polymerase here can come in and bind to the promoter and then go down the DNA and transcribe these genes. And then as a result of that, you're going to have increased production of these gene products, beta-galactosidase, lactose permease, and galactoside transacetylase. And then as a result of that, the cell can more efficiently transport glucose into the cell and metabolize it. So in this case, if glucose is not present and lactose is the sole metabolic substrate, you're going to have increased cyclic AMP because you don't have glucose blocking adenylate cyclase. So increased cyclic AMP, increased binding of cap to the cap site, which facilitates RNA polymerase here, which will then further increase transcription of these genes here. And again, you don't have this repressor protein coming in and blocking transcription. So you have both of these mechanisms, both the cap binding to the cap site and then the repressor protein mechanism. Both of these are working in favor of transcribing these genes so that you can carry out more efficient metabolism of lactose. And that makes sense. You don't have glucose available. So you need to have lactose metabolism operating at its peak efficiency because the cell's metabolism depends on it. So a different scenario here, if glucose and lactose are present, then you're going to have, obviously, adenylate cyclase is going to be inhibited by glucose. You're going to have decreased cyclic AMP, decreased cap binding to the cap site. You're not going to be able to facilitate this promoter binding as much by RNA polymerase. And then glucose is going to be used until it's no longer available, and then the cell metabolism can switch to using lactose. Now, since lactose is present here, you're going to have some of it still being converted to allolactose, and this repressor protein will be inactive. So you don't have this binding to the operator, but also since glucose is present, you don't have this cap either. So it's kind of like a stalemate. One mechanism is working in favor of these LAC genes being expressed. However, since glucose is present, you don't have this cap binding site mechanism working in favor. And that makes sense because you have glucose present. You don't need to ramp up lactose metabolism just yet. Now, what's interesting is you have both these mechanisms working at once. You don't have the repressor protein binding, but at the same time, you don't have cap binding either. And that makes sense. This is going to favor glucose metabolism, which is what the cell wants to do in the first place. It wants to use glucose as its primary metabolic substrate. However, the gene is ready because it has some lactose present. You're not going to have this repressor protein bound, and so when glucose is used up, the cell can easily switch to using lactose as its main substrate. When glucose is used up entirely, this is going to switch, and you're going to have increased cyclic AMP and increased CAP. All right, so now to summarize these scenarios, just so you have these down and you're ready for your test. If you have decreased glucose and lactose is present, you're going to have increased expression of Lac Operon genes. So this is the ideal for, in the world of expression of the Lac genes, this is the ideal scenario, right? You don't have glucose, so you're going to have increased cyclic AMP, increased CAP binding to the CAP site. That's going to increase binding of RNA polymerase to the promoter, right? You also have lactose present. Lactose gets converted to allolactose, which then binds the repressor and then renders this inactive. So then there's no binding to the operator. This doesn't happen. And RNA polymerase can fully go down the DNA and transcribe these genes. Now, if you have increased glucose and lactose is present, what's going to happen is that you'll have glucose here, which will block adenylate cyclase. You'll have decreased cyclic AMP, decreased CAP binding, decreased binding of the RNA polymerase to the promoter. However, since lactose is here, you're going to get some allolactose. It's going to come in and bind this repressor, and you're going to inactivate this repressor, so you're not going to have this binding to the operator. So as a result of that, you'll have a basal level of expression of the Lac Operon genes because you don't have this repressor protein blocking it. Even though you don't have CAP, you're still going to have some RNA polymerase binding to the promoter and some transcription of these genes. It's just not going to be at peak efficiency because you need CAP for that. And the reason you don't have CAP is because you still have glucose. Now, if you have increased glucose and there's no lactose around, you're going to have no expression of Lac Operon genes because, again, you have lactose, decreased cyclic AMP, decreased CAP. This doesn't happen. And then on the flip side, you're not producing any allolactose, so this is not happening. And then the repressor protein is fully active and able to bind to the operator and block RNA polymerase. So both these mechanisms are working against transcription of the Lac Operon. And so that makes sense. You're going to have no expression. And then lastly here, if you have decreased glucose and no lactose present. So first of all, if you have no glucose, you'll have increased cyclic AMP, increased CAP. But if you have no lactose present, it doesn't matter. You're going to have no allolactose. This doesn't happen, and you're going to have the repressor protein active and binding to the operator and blocking transcription of these genes. So you'll have no expression of the Lac Operon genes. And that makes sense. If there's no lactose around, there's no point in expressing these genes because there's no use for them. So this summary here is really important. When you encounter a question on a test that is asking about the Lac Operon, you really want to pay attention to where the glucose level's at and where the lactose level's at. Because as you can see in this diagram, this is going to dictate the regulation of these genes. And so by paying attention to that and understanding how the level of glucose plays into the regulation, how the level of lactose plays into the regulation, you're going to be able to figure out the answer to what they're asking, which is going to be something along the lines of how much of these genes are expressed, which is going to be along the lines of what level of gene expression is occurring for these genes here. Is it none? Is there somewhat of it? Is it fully up? Is there no expression? Is there kind of a middle level of expression, or is it fully expressed? And these really aren't that hard. You just got to kind of draw this out or think it out yourself a few times and you'll have it down. All right, so over the next few slides, we're going to talk about regulation of eukaryotic transcription. So this first level of regulation we've actually already talked about in the first lecture when we talked about chromatin structure. So you have euchromatin, which is shown by this diagram here, and you have heterochromatin, which is shown here. And this is all about the physical access that RNA polymerase has to DNA. So in the case of euchromatin, you can see it unwinds, and then RNA polymerase has a lot of access, and other transcription factors can come in and access the DNA and carry out transcription. And then heterochromatin is densely packed and allows little access to DNA and limits gene expression. And remember, this can go between these two states. So it can be heterochromatin, cell needs to express a certain gene, so then it unwinds into euchromatin, express the gene, get the protein, and so on. And so this is a fluid state between these two. It just depends on what's required in the cell at the time. Now at the DNA level, there's actually sequences within the DNA adjacent to the gene and the promoter where transcription factors will bind, and these can have impacts on gene expression. So first you have enhancers, which are shown here in blue, which is next to the promoter here, and transcription factors, which are TF here, can bind and then help facilitate binding of RNA polymerase to the promoter, which then helps with getting transcription started. You can also have what's called silencers, which are sequences of DNA where other transcription factors, called repressors. Transcription factor is a very broad term, so you can have it where they bind enhancers and facilitate. You can also have it where they bind silencers and repress it. So in this case, they bind the silencer and they block RNA polymerase from binding. So this doesn't happen and lowers the level of gene expression. And then lastly, we've shown you here in this diagram that the enhancer and the silencer are adjacent to the promoter, adjacent to the gene. And there are situations where this is the case, where it's all just one linear sequence here. However, there's also situations where you have the gene and the promoter down here, but then way down the sequence are the silencer or an enhancer or both. And even though they're not directly adjacent to the promoter or the gene, they still have an impact. Because the way the DNA structure turns, it actually brings the enhancer and silencer in close proximity to the promoter and the gene, and thus they can still carry out an effect. So even though in sequence of the nucleotides, they're not directly next to the promoter, because the way the structure turns, when a transcription factor binds here or here, it's still in close proximity to the promoter and can impact RNA polymerase binding. All right, so now some clinical pearls to wrap this up. So first, tamoxifen, and this is an example of targeted chemotherapy. Now remember, we've talked about cytotoxic chemotherapy, which targets DNA replication and rapidly dividing cells. This is different. This is the newer age type of chemotherapy, where it targets a specific molecular driver of cancer within the cell. And in this case, the molecular target is the estrogen receptor, or ER. Now you may have heard of ER positive breast cancer. And this is where the estrogen receptor is one of the drivers of this particular breast cancer. And just to review, estrogen receptor, it's a steroid receptor because estrogen is a steroid. So estrogen crosses the cell membrane, comes into the cell, it binds the estrogen receptor, and then it forms this complex of estrogen and the estrogen receptor. Then this complex of the estrogen receptor and estrogen translocate to the nucleus and help facilitate transcription of certain genes. Now when you have tamoxifen, so tamoxifen, again, is a targeted chemotherapy, and the target here is estrogen receptor. So targeted chemotherapies, they'll target specific molecular drivers of a specific cancer or a driver that's involved in multiple types of cancer. So in this case, tamoxifen comes in, it binds the estrogen receptor instead of estrogen and blocks, so it essentially blocks estrogen out. And then it forms a complex with tamoxifen and the estrogen receptor like this, and then hence no DNA binding occurs. And so it's not able to go on and facilitate expression of genes down here. All right, and lastly, we're going to finish up with talking about microRNAs or miRNAs. So what these are non-coding RNA sequences within introns. So you can see here we have our introns. There are these gray portions of the mRNA strand. And then you have a sequence within one of the introns that's a microRNA. And these are involved in post-transcriptional regulation of gene expression. So what happens here is you have splicing that occurs. You splice out the introns like this. You connect the exons together to form your final mRNA that it then undergoes translation. What happens here is you have this microRNA that then can complementary base pair with a sequence within the final mRNA transcript. So in this case, it's in exon 3. Part of the sequence of exon 3 complementary base pairs with this microRNA. This creates a problem because it can result in inactivation of this mRNA or even degradation of the target mRNA. Because by binding to here, you could potentially block the ribosome from carrying out translation, which then creates a problem because you can't just have a final protein with just this part of the sequence. You need the entire sequence to form the final protein product. So it could result in decreased translation. And the other thing with microRNAs, it could also lead to cleavage of the mRNA. So you could have enzymes that cleave the mRNA and cleave this into two strands, which doesn't help anything either because, again, you need the entire sequence to express this gene. And then the other thing is if it doesn't get cleaved, it could be destabilized and then eventually degraded. Now this comes into play in cancer because some cancers involve abnormal expression of microRNAs that silence mRNA from genes that specifically halt cell division or promote protein degradation. Now you have tumor suppressor genes, which we'll talk about in much more detail in the cell cycle lecture. Tumor suppressor genes keep DNA replication and cell division in check. If these aren't around, then it goes unchecked and that's where you can develop cancer. The other thing is proteasome proteins. These degrade proteins over time all throughout the cell, but where this comes into play is they'll degrade cells that, again, are involved in DNA replication and cell division. Because, you can't just have these processes running wild, uncontrolled. That's how you end up developing cancer. So if you have decreased expression of genes that code for either of these types of proteins, then that's where you get problems and that's where you can get cancer. So where microRNAs come into play, so let's say this microRNA is, base pairing with this portion of this mRNA, and let's say this mRNA is coding for a tumor suppressor protein. Well, if you don't get translation, if this gets blocked or if this mRNA gets degraded and you get decreased expression of the tumor suppressor protein, well, then that's going to result in cancer. All right, so that wraps up our discussion of transcription.